Good morning, good afternoon, everybody. Welcome to day 2 of TMF Week, session 2, day 2. My name is Oliver Pierce. I'm the Director of Commercialization at Montrium. If you're a frequent attendee of Montrium's webinars and sessions, you'll be all too familiar with me. I have a few slides to share to kick this session off, but I wanted to first, as people are falling in, let us know where you're dialing in from. Are you coming in from, so Aviv here, coming in from Montreal. Hello, Aviv. Where are you coming in from? Are you coming in from the United States? What city, what country? Let us know where you're joining us from today. I wanted to do a quick reminder. TMF Week obviously delivered by Montrium as a technology provider would not be possible without all of the friends, as we like to call them, that have been a part of the event so far. So our speakers, our advisory board, all of our participating organizations, it would not be possible without your involvement. So thank you so much for all the commitment to putting on such a great event so far. We are now sort of halfway through day 2. We've got lots and lots more content to share with you and lots of amazing speakers. You'll hear from over 40 different speakers over the course of this week, which is phenomenal. And there are over 25 different sessions for you to attend, all free of charge. So today's session that I will be hosting is the foundation of the clinical trial story starts with study startup. Really great presentation. We're going to be online for about 45 minutes. There'll be roughly a 30-minute presentation, and then we'll have about 15 minutes for questions at the end. So please throw your questions into the Q&A panel. The Q&A panel should be in the top right-hand corner, in between the chat and the polls. There will also be some polls that we will be asking the audience for engagement throughout this presentation, so our speakers will be launching those. Again, you can access the poll questions by going up to the top right-hand corner and selecting polls, and you'll see all the polls there. All of the sessions that you attend at TMF Week or don't manage to attend will be recorded. So don't worry if you end up having to leave to go to another meeting. All of the sessions will be available after the session, as the day preceding the session. Every single attendee of TMF Week will receive a TMF Week certificate of attendance for the total event, not individual sessions. We do have networking breaks, so for those of you that had a chance to network in between the 2 sessions this morning, we do that after every single session. We have a longer networking break after today's session, so we have a 45-minute networking break coming right after this, where you guys are able to join different tables of discussion. So we have tables on the TMF Reference Model, we have tables on ICH E6R3, we have a number of different tables and topics where you can join and talk about different subjects. So today I have 2 speakers that are going to be joining us. We have Elvin Thaland, who's the Director of Industry Strategy at Oracle Health Sciences. Elvin is a recognized industry expert in clinical trials and has over 20 years of experience working as a clinical business analyst consultant at major pharmaceutical companies, including Hoffmann La Roche and J&J. He is the co-chair of the TMF Reference Model Exchange Mechanism Standard alongside Montrium's own Paul Fenton. And he's also earned a Master's of Science in Industrial Engineering from Aalborg University. And Elvin is joined by Jennifer Wilson, who is the Director of TMF Operations at Syneos Health, a seasoned project management professional with over 20 years of industry experience and over 18 years actively leading domestic and international clinical programs, having exposure to all phases, including orphan drug indications, rare disease and gene therapy clinical studies, and complex study designs. Jennifer has therapeutic expertise in oncology, respiratory, infectious disease, CNS, metabolic, gastroenterology, vaccines, hematology, and pediatric clinical trials. So lots and lots of experience on today's call. So I'm going to stop sharing my screen, and I'm going to add our illustrious guests today. Welcome both. Elvin, you can begin preparing your slides. I will stop sharing my screen.
Yeah.
And we already have lots of questions.
Okay.
And I'm going to add you to the screen. I'm going to remove myself. So Elvin, Jennifer, I'll hand it over to you. Good luck.
Yeah, thank you, Oliver. Okay, and so here's the agenda for today. So there's sort of three things we'll go through. First, where does study startup actually fit into the bigger picture here, the full picture of doing clinical trials? And then after that, I'll talk something about the study startup and how, at least from an operational point of view, it's all about speed. And then go into lastly into some detail about the complexities of site activation. And then, get back to Q&A at the end. And with that, we'll start into the first one with where does it fit in the bigger picture. So if you look at it, the bigger picture is the program, because in the end what we end up filing is generally, it is the program to get the market authorization. So it starts out with from a clinical point of view, that you start with the program approval. And at that point, you're now getting your approval for doing the program, the protocol itself for the study. Followed in the US by an IND, so you can go into human for testing. And then it have normally three distinct phases you go through that often have been separated into individual protocols, but more and more get combined. So you have your first in human, you have your proof of concept, and then your pivotal phase 3, where you cover all the areas of what you intend the indication for that drug to cover when you go out for your market authorization with an NDA filing in the US. If you then take that down on the study level, which are more where the clinical trial master file operate, there you go in with, you get an approval, you have your title normally, you get your synopsis, and then you get a protocol approval, you get through your enrollment, you get your data back for interim analysis, you do clinical closure, and then you have your final clinical study report. When we get into the true study startup area, that's where you sort of select your country, identify where do you actually want to execute your trial, and when and you get it activated, you enroll it in clinical closure, and site level is similar, more that the country is the regulatory area you do it in, and we'll get into that later. But you select a site, you activate the site, you enroll them, and when you enroll, you execute, and then you end up in clinical closure. And the most important participant in the study, often in our systems called the subject level, more and more mentioned as the participant. And they enroll, and then they generally go through that data is eligible for the study, and then feed into the interim analysis. So this is the bigger picture. And if we then think about where does the study startup fit into this? Study startup is, as I mentioned before, is when you sort of have gotten your financial approval for doing the study. Now you create your synopsis, and you can see it covers a lot of milestones. In the bigger picture, it actually covers most of the at least planned milestones and decisions. So from a behavioral point of view, when you're doing study startup, you move towards your decisions and your events. So you have planned events and decisions, and then you collect your artifacts, your TMF artifacts towards that. And when you get them, hit each of the milestones, you check them off, or you take a decision. So those are sort of the behavioral area of study startup. If you then compare with study execution, so the execution comes into when you have activated your site, meaning the site is ready to enroll. And that definition can vary from sponsor to sponsor and even CRO to CRO, where you actually say activation, but it's very related to IP release. And IP release is a regulatory term and always the same. And then it ends at clinical closure, which are normally when you do the last site payment, which means you basically have ensured you got everything squared off at site. And then the last one, and at least, is the study reporting, where it starts when you start getting your data and you get your interim analysis out, and then end with you have your final study report. So that's sort of the bigger picture. And going back to study startup, so what is it from a TMF point of view? And this statement here is from ICH E6R2. specifically related to section 8.2, where this is a study startup is about permitting the evaluation of the conduct of the trial and the quality of the data. So it's ensuring that we are ready to go fundamentally, and we can deliver quality data. And then we also ensuring that the actors, sort of when we talk about the story, we need to have a cast, and the cast and the compliance of the cast, the investigator, the sponsors, and the monitors, and that they follow good clinical practice. And if you talk about the changes coming in R3, I'm pretty convinced that this statement here will stay pretty much the same when we talk about that, because the purpose will still be the same. This next one here, though, will likely change some, because in the current R2 version, we have a list of documents which also reflect the TMF reference model, which likely now we get into R3 will be replaced. Instead of having a list of documents, the checklist that Thomas also spoke about before, will go into more having risks that we mitigate with this. And with this, we go into our first polling question. So Jennifer, I don't know if you can.
Yes, let me launch this.
So I can read it out while you're launching it there. So
Yeah.
when do you begin assessing TMF completeness? Do you do that when you approve your protocol? Do you do it at IP release? Do you do it at the end of the study when it's over? What is TMF completeness? So those are the options. You see, I think it's live.
Yep,
So
you should be able to. Oh, good. I'm getting some numbers here, so we'll give it just a few more seconds for people to
when do you begin assessing TMF completeness? Do you do it when the protocol is approved? Do you do it at IP release, basically when the first site is activated? Do you do it at the end of study? Or, what is it? So we'll give it just a few
Okay,
more.
5 seconds, and then I'll share the results with everyone.
Yes.
Okay. Okay, so a good mix here. I just want to see that it show results. Here we go. So I think you'll be able to see on the screen. Can you see the result on the screen, everyone? Or maybe Elvin, can you see it?
I can see it, and for any participant, for them to see it, they have to navigate to the poll tab at the top on the right. So they can see it there. It won't pop up on the shared screen, but they can see it if they navigate over like we do.
Okay, sorry about that.
No,
so you can see there's a little bit of a mix here. Certainly at approved protocol took the majority of the findings, the polling question. I'm a little nervous about you guys who wondering what TMF completeness is, but hopefully by the end of this week, you'll have a very thorough understanding of what TMF completeness is. Definitely, we want to make sure we're thinking about TMF completeness really at the very beginning. And even I would argue, perhaps there's one other category that we didn't put in this poll, but we should be thinking about, and it really goes back to Elvin's slide on, really understanding the clinical development plan for your asset, right? For the product that you're investigating and look to launch and put out into the marketplace. We know a lot of things about that drug or medicine when we're really just kind of thinking about our campaign for this in early trials, right? So if we're in our phase 2, phase 3 environment, there's a lot of documentation that we probably already have that we can think about making sure is included in the TMF, assessing that it's complete and ready for that end product in the TMF and as a holistic view of the study that's coming. So really great answers. Thanks for participating, everybody. And I'm catching up with you guys who are wondering what TMF completeness is. I'm coming to you.
Good. I think with that, so I don't know if you have more comments on this discussion slide here, Jennifer. So what do you think? When can you assess TMF completeness? I think that's
exactly.
the other side of the question, right? So when you do it, when can you do it, right?
For sure. So as we think about a study, so as we begin our study, we definitely have some early indicators. We tend to get things moving out of the gate really quickly. As Elvin mentioned, study startup is always the focus for speed and getting sites identified, getting contracts in place, those types of activities. But really, as I said, there's some early things that we could be thinking about, right? Is this a study that we have a comparator product? Do we have the documentation on the comparator product that's going to be part of the trial? Can we look to file those things even in advance of the study synopsis or around that time? So there's definitely some indicators that we should be thinking about that often, tend to be overlooked when we do have such a focus for site identification, contracting, getting these people really engaged at the very beginning. There's a lot of documentation we can definitely get from those early days, before study synopsis, right at that area. And then certainly as your final protocol is, your protocol is finalized, you have a lot more firmness to exactly the documentation that you're going to be expecting. And I know we're going to talk a little bit later about expected documents and how to be thinking about those from the very outset, and making sure that you're comparing expectedness to what's actually in your TMF at certain points along the study path.
Yeah, very good. Exactly. We'll get into that here. And I think with that, we'll thank you, Jennifer, move into the next section. So it's all about speed. At least from an operational point of view, this is study startup is where you can make a difference in how your timelines are. When the trial is first going, it's more about keeping the train on the track and getting there on the expected time. With this, we actually go into the next polling question. Jennifer, can you launch that one?
Yes.
So which percentage of your TMF reference model artifacts? So if you look at it from the TMF reference model as we know it, which percentage do you actually think is collected before the site activation? So is it less than 20%, 20 to 30%, 30 to 40%, 40 to 50%, or more than 50%? I think we launched a different poll.
Yep. And sorry, Elvin, I don't see the poll that you have here. I don't see it in our listing of available polls on the side of our screen. So I apologize for that.
No,
but I think what we can think
of I was the one trying to get them up before. Somehow I don't even. Okay, interesting.
Right. So we can probably think about this just in terms of artifacts in general. The question that you have on the screen is around ethical approvals, which we did have a little bit later in the presentation. So apologize for that, mix up. But just as we think about artifacts in general, I think that there is some discussion we can have about the numbers that you have on the screen here, Elvin. We'll see what everyone sort of thinks about with the site ethical approvals specifically. I'll share that result right now. And I will show result to everybody.
And I think we should, though, we can talk a little about it because it's later there, but we shouldn't cover it too much here. I think with this one here. Yeah. Sorry.
I think really the point I just wanted to make is, in response to the poll that Elvin has on the screen here that you can still see this less than 20 all the way through greater than 50, when we think about the artifacts involved in study startup, we do see actually a tremendous volume. And in fact, it is greater than 50% that we have artifacts in the TMF that are related just to the study startup items and classifications and documentation that we get in to support the site activations and study startup pieces. We'll talk a little bit more about the results when we do get into that side of the presentation a little bit later, and apologize for the technical issue on this polling question. But I think your next slide, Elvin, talks through a little bit more detail about the extent of the artifacts included here.
Yes, exactly. I just switched to it because so in reality, it's actually 53% of the TMF reference model. So 131 of the artifacts are of the 249. That's the total of the TMF reference model in the latest version. 131 is based on the milestones. If since I think 3.1, we had milestones in the TMF reference model. And if you select anything that's basically numbered up to 3, so 1, 2, 3, which are study startup components, they are related to study startup. So you actually get the first version, at least, of all or over half of your artifacts will already exist from a TMF reference model point of view at the time when you start the study. And from a volume point of view, I argue it's actually generally more than 50% from a pure, when we talk about documents for sure, that you have already before you start your study. So from a completeness point of view, you already have over half of your documents existing when you start your first study. Because volume actually increases as you go down. You will see informed consents and so on, depending on the number of patients, be very large when you go down. And of course, we have a lot of safety data and clinical data, but most of them are not documents per se, they are data. So yeah, that's what we normally see. And with that, I would actually go into, this is a history, and the interesting thing here, when I started 10 years ago with a company called Globalto at that time, we went into study startup, and we went into this totally green field. And by chance, it was actually seniors at that time, INC, we did it with. So it's just by chance, Jennifer and I are here together today. And 10 years ago, all that existed was Excel in study startup. And if you look about it from running a trial and especially starting up and collecting all your documents before you even activate your site was all in Excel. Even if you did a study that was 70 countries, a phase 3 trial, it was all Excel, and they were all different. All over the world, the format was different. So it went into how can we align the format of these Excel? How can all countries agree about what they track, what are the milestones, and later on actually going down to the document level, what are the document and file names that go into study startup. And one of the elements I want to bring in here is how we look at how we move along. And when we talk about speed, it's about getting there on time. And we are familiar with that. So if you think about, we have these navigation apps already, and it's all about how can you're used to using this on a daily basis. So it would be great if we actually could have the same view on when we do a trial and how we do study startup. So this will suggest the best route to get there. You can choose a different route here if you have better information than the system. And then it will give you information about current bottlenecks. You can see highlighted, of course, in red. So this is how we actually used to navigating out in the world out there we now are getting allowed to go back to after the pandemic. And if we now go in and study startup and look at that similarly, there we first have to understand what are the more underlying component of study startup. And to do that, we'll zoom in on those milestones we talked about before. So the first component there is the study definition component. And this one, historically, you could say have been very stringent. We have synopsis, we have a study package, and we have the protocol approval. But these days, this becomes much more agile because you learn much more throughout the whole study startup before you actually define what the protocol is. The first component is the site the selection, and the site selection and country selection for that matter too. But if from a site point of view, it's the site selection where you identify the site that fits your study, and then after you identified enough sites that should be sort of the pool from a feasibility, you go through that, then you go into your activation component. And this is where you ensure they actually meet GCP. So you go through all the content that need to be done, and you do your contract negotiation. And then after you activate the site, you enroll. And when you have enrolled all the patients you need to meet your study, now your study is up and running, fully started up. So these are the sort of high-level component of study startup. But let's take that now and can we think about that as a map? And this here is from a site level. So first here, what over these decades, and I've gone through and identified what are the counties. Think about the counties on a map and think about them as functional areas. And generally, these functional areas, what is also, if you use a CRO like Cineas, that you would outsource into from a functional outsourcing. And on this map, you can also have your cities, or we use them as milestones. So these are the points you want to get to as you go along. And then you have the roads out there, the roads you can take. And now you suddenly have a map where you can start measuring your cycle times and even your quality at, because these milestones are also key elements to when you can ensure your TMF quality. And if we go in, where do you want to go? And if we take in this case, we want to activate a site. So this is your target, that's your endpoint. And if you start at your country selected. And what is special about the startup, you select your country, and now you have to take your road, you have to get all your sites selected, you identify them, you select them, you get all your infrastructure, you have your investigator visits, you activate, and you also have to do the site qualification. So that's the contracting, essential documents of all the actors. You have your ethical approvals, you have your country approvals, your import-export license. You have to have your IP ready to go. And then you also have all your technical infrastructure, EDC, IVRS ready to go. And now, in this case, different from when you just navigate in the real world, here you must take all the roads. And with this, do we actually have, yeah, this one we have still the polling for. So somehow we have 4 polls in. I'm not sure what happened there. If we go this
okay,
one.
so you have the polling section on the top tab there, everyone. You can answer the poll question.
Yeah, and how long do you actually think a study startup takes on average? So this is an industry, coming from an industry survey that was done back in 2017 by Tufts. So we can say, so how long do you think it takes? Less than 5 weeks, 5 to 10 weeks, 11 to 20 weeks, 21 to 30 weeks, or more than 30 weeks? And lastly, I have no idea. I hope you'll get something at the ones where you have an idea, but try to pick one of those. And see it all coming in.
Yeah. We'll do 5 more seconds and then I'll end the poll. Some good variety to discuss here, Alvin.
Yep.
Okay.
So, you want to read it out? What, like, so 30. So if you take the 2, it's over 50% comes out between 5 to 20 weeks. With it's 55%. So the good portion laid less than 20 weeks. And so it's between 5 to 20 is the majority, but even a good portion up to the 30 weeks. So ballpark less than 20 weeks, I think most people think it takes. And with that, I'll actually go in and we'll look at what is the reality. So this is, as I said, from a Tufts study that was done back in 2017. And this is for a new site. There's also some timeline for recurrent site. But if you look at here first, the first component, if you look at about site identification, that alone from the industry takes 6.5 weeks. And the site selection component, meaning normally feasibility, takes around 8 weeks on average. And then the actual activation component takes 22 weeks. So that's a total of 36.5 weeks are the average it takes to activate a new site. It's slightly faster if you have a recurrent site. And this is an industry standard. And since this survey, I know Tufts had done it again. This one we actually Gobalto sponsored at that point when they did it, but they have done it in others since then, and it actually now takes longer. So timelines have actually gone up and not down, simply because trials are getting more complex, and we are competing more about the same sites and the same studies. So it actually takes over 7 months to activate a site on average. So, and of course, from a sponsor point of view, I must find my patient and enroll faster. It's all about speed. And then from a CRA, how can I find patient and how can I enroll faster? And a patient, don't forget the study participant. How can I wait 7 months to get into a study? Which are reality from a site is contacted in the beginning, generally it takes up to 7 months or longer before they're up and running and actually can get into the study. So with this, if we now go in and look, how does that look compared to a TMF and how we operate there? So what are the key decisions we go through? And if you look at the identification, the first point, and you can see here now I put in sort of the taking the trip and telling the story, the TMF filing. The first study relationship of a site is when it sort of get identified. And that means you now have decided you want to start visibility on the site. So you start in identification out from your black book or whatever to a find site, but as soon as you have done it, you create a site, and normally that's in the CTMS. You go in and create it and you get an ID. So this is the first occurrence where a TMF would know about it. And then you get to the decision point, do I select this site for participation in this study, or do I not? And one option is you say, no, I do not select from a feasibility, which you should also document, that's TMF records. Or in a, of course, preferred mode, you select them, which are a key event in the whole process. And next you go into the site activation component, where you go through and say, okay, they're qualified, they're essential document, and you get the contracting. And then you set up your infrastructure, both technical and non-technical infrastructure. And there you have your non-technical infrastructure you need to document and get into the TMF. Again, another milestone, a decision. You have your systems you get up, EDC, IVRS, safety system are ready to go. So all that infrastructure needs to be documented. And then the qualification itself, your regulatory approval, your essential documents, 1572s, and your contract are key elements. And all these happen long before you actually do your activation. And then you get into your activation with your investigator meeting, your SIDs. And here, you have your IP release, probably the most critical one that we all know when we, from a regulatory point of view, we now hand the drug over to the investigator because we have qualified them to actually do the study. And then you activate. And activation means fundamentally now this site is ready to enroll the first patient. And then finally, you start your enrollment. And this element is often not looked at as a TMF event, but it's very critical to have a successful study, that you actually have the patient needed to meet it, because that's the critical one for what time you'll end on when you have all your patients enrolled. So sort of this is the bigger picture here for this enrollment. And Jennifer, what do you think from this? When should you file? Are this more these points? Is this the right level?
Yeah, and I, you know, Alvin, I just really love the analogy that you put to this map on the page. So, you know, looking at the counties, the cities, the roads we're taking, the navigation we're doing, and you know, if everyone sort of thinks about these TMF, you know, points that you have included here, these are really like the opportunity for us to sort of like check the gas meter, right? What do we have in the tank? What do we have in the TMF? Do we have everything we need in there at this particular milestone, right? We think about milestones, we develop systems and technologies for electronic TMF systems that we use that, you know, incorporate milestones, but at the end of the day, they're not, you know, they can't be 100% automated, right? We have to as well be thinking about what other documents do we have. Some studies that are very, you know, a little bit further along now, but earlier genetic-based studies, we didn't have these kinds of things known in the TMF reference model of what types of documents to expect. So we were laying down groundwork. There's always evolution with the TMF model. So we have to be thinking critically at each of these milestones, in addition to using the technology that we have to help us and guide our thought process, but it still is a little bit of a manual effort. We need to be critically thinking about which documents are generated along these points and really making sure that gas tank is full before we keep going, right? We can be inspected and audited at any time. So we have to make sure our TMF is, of course, contemporaneous and complete.
Yeah. I think that there have been this tendency first to file around IP release and activation, because this is the first time you actually have patients in the study. So historically, that's often when it had been done, but there's actually many milestones that go before that where you can assess completeness. I think in point of time, we probably have to move into the final section here. So now with that, I will actually go into some of the complexity of this study sitemap. and I'll focus on the selection and activation compliance component, which are often where CROs are involved as heavily in these areas. And you can drill into this, and there's often even more milestones. So you can even argue, and some of these milestones is where you should possibly even do your checks and check your gas tank, right, again, and go and check your oil and see you're all ready. Where you have these key protocol approval, you get your study package. When you talk about the qualification, a lot of it is getting the right study package. From the study level, if it's a sponsor CRO scenario, sponsor will hand over their study package, and the CRO will start going through it country by country. And after that, site by site, you get your site ID, you sign your CDA, you get it selected, and then you go through all your approvals, your profile, essential document collection, your contracting, and then in the end, you can say your site is qualified. So these are all key milestones that occur throughout that. And an interesting thing is, you will often have your CDA signed before you have your protocol approved. So many times we try to plan and measure from protocol approved, but you actually have more important information on a lower level like the CDA signed. And with this, what we want to go in and focus on one key element, the regulatory approved essential document. We'll try to move along fast here. So first here, I know this is tiny, but it's just to tell something about the complexity and the differences that exist and why it's so hard to move from an Excel into actually a system when we talk about study startup. Because if you take a country like Thailand, all the dependencies of different regulatory bodies involved in your approval. So in Thailand, you have your local site IRB, you also have your import license you have to do, and then you have a separate regulatory body that first can start. So you have multiple different submissions you do. You have one for the site, and when you're done the site approval, then you go to your regulatory approval for the study. And then you also have a regional central ethical component in it. In Argentina, on the other hand, you have other kinds of dependencies between the regulatory. You have central, and you have your site. So you have 3 different levels because you have a regional one in Argentina, plus you have your import, drug import. In the UK, especially after Brexit, they'll still be separate. And we don't even have now the European, at least the European will harmonize, but still have multiple countries that need to organize on it. If you look at the British one or the UK one, we have the Central Ethical Committee, we have the NIH, and then you have a regulatory body, and you have the import. When in the US, you generally don't have a regulatory body, and when you have your IND, that's that component where you have a central, and it's more and more moving to central simply because of speed and consistency and avoiding protocol amendment. And then you have your site. If you have an academic site, you often will have your local IRBs. So that's this combination. So it just shows that's a vast difference. And if you have 70 different countries participating in the study, you have all these different kinds of approvals. And if now we can go, we already did the polling question, so we can gain some time here. So I don't know if just read out the answers here. I think most of it fell into between 10 to 20, 40% said 10 to 20 to this one, but more than 36% said more than 20%. But I would say if we put a number on there, around 15 artifacts was sort of the average, if we should say that. But what is reality? And here, what I've done here, and this is one I did some years ago with the Essential IRB as an example based on their submission form. If we take what goes into first the submission, later on we'll go into the approval. So there would be a questionnaire, and we can map that into what I've done is number the TMF artifacts underneath, 03, 01, 01, which are normally the regulatory submission artifacts. And then you get all the study material. And when you submit, you have the protocol itself, you have the ICF, and then you have all the subcomponent of the ICF, which can be study dependent. So depending on what's required, you would have all these underlying ICFs need to be there. And beyond the ICFs themselves, you have translation. So if you this is in a case in the US, depending on the site, you can have a Spanish version, you can have a Chinese, Portuguese translation version of that. And then you need all your product material. So the investigator brochure that tells about any safety issues, you have your package insert that go out, you have the recruitment material, so you can get your study participants. So that would be a brochure, ads, flyers, telephone scripts, and study questionnaires. And then you have the patient material, what you hand over, so subject diary, the subject questionnaire, subject information sheet, subject participation card. So this is all the information you need to submit for approval in the US. And interestingly enough, the material generally from country to country, what you need to go approve is usually the same, independently of country. How you do it is very different. And then on the other hand, then from a site point of view, you also have a site-specific questionnaire, which would be related to 04, 01, 01. And underneath that, you have your principal investigator material. So their CV, their med license to support that they're qualified. And if you have multiple satellite sites, you also need to file that separately. And then you have site-specific recruitment material that's related to anything that's local for the site. Again, the brochure and all of that. You have the study material, the ICF, so site-specific content. If they need a bar card here in San Francisco or something like that they need to consent to. And then you have the translation of that content too. So this is the picture of what a submission entails and all the artifacts. And generally, we just deal with that as two artifacts in the TMF reference model and don't really track, at least in the TMF, these individually. And if we now go back to the approval, you have your study approval letter, so you get an approval letter back. And with that, you get in that letter approval of all of this content. So it's one artifact, but in reality, it's reflecting a lot of individual artifacts. And you also get then, if you talk about the recruitment material, you get that back separately, and they're generally stamped, meaning that's the original with a stamp that this is okay. So that's a stamp on it. And from patient material, you get the same for the diaries, the questionnaire, information sheet, all stamped material coming back in the same approval. And on the site side, you get the letter back, and in there you'll also get the investigator approval, hopefully, or partial approved, because it's not just a binary decision, approved not. It could be partial or conditional approved. And then you have your ICF, and then at the end you have your recruitment material, and which are stamped. So the answer is, if you look at it purely from a TMF reference model, it's actually over 26 artifacts, which by document volume is much, much higher than that, of course. So, Jennifer, what do you think? Should all of these be filed as individual artifacts in the TMF, or how should it be filed, or just the 4 that is in the reference model?
Right. So it's a very interesting question. I think there's probably some discussion to be had on that. We submit the documentation, we can clearly see what's been submitted, and we have requirements to file those things. But we don't necessarily think about what the ethics committee or regulatory body has done to modify those things and provide something differently. So we have to be very carefully looking for those changes, making sure we file final copies of these things after they may or may not have been modified through the process, right? It tells the story much better. We make sure that what we submitted has indeed been approved. How do we do that? Do we evidence, in the letter? Is there the documentation that maybe has a stamp on it from the regulatory agency or ethics body for us to evidence a clearer indication of approval without having to sort of navigate through just the letter or just the submission information? So I'd argue that there's probably a need to be very carefully looking at what is being provided as outcome documents from, following the review and filing those very separately.
Yeah, I think it's definitely an area where the reference model itself could at least, I don't know if sub-artifacts is one option to file these individually, so they're like an approval sub-artifact that's tied to the individual artifact, because these will exist as individual artifacts already. But how do we actually track the approval of these, the submission and approval of them, right, in the TMF? And I think that at least the reference model itself has not answered that generally. I know we are tight on time here, so if we need some Q&A in, we should try to move into that, I think.
Yep. So I have one question that I received through the chat. Thanks for everybody who submitted. If we don't have time to answer questions entirely today, please keep them coming. We will make sure that we do get back to you. We will try to answer all the questions that we receive here. But one of the questions that really stood out to me, Elvin, was a question that asked about how we measure TMF completeness, right? What does that mean? How is that defined, right? And I think in our industry, that's often something that really evokes a lot of discussion, right? Is it just the number of documents that we're counting to be included in there? How do we think about it? So my interpretation of that, really, that I try to stress and implore to people is it's a balance between quality and quantity, right? We might have all of the documents included in our TMF if we're looking at what we expect versus what we have filed, but then it becomes, how do we think about it in a cross section, right? My example that I often provide is, if we have a monitoring visit that has happened where we generate a visit report, well, there's so many other artifacts that go along with that, right? We have maybe a confirmation letter, maybe we have a follow-up letter, and then we have further follow-up to make sure we've closed those actions. So it's really about telling that entire story, right? From end to end. So it really has to be a matter of assessing for completeness in terms of document count versus what we expect to be filed, and the quality of the story that we can reconstruct, right? I think for anyone who's on this call who have participated in inspection or audit, it's very, very clear that's the expectation of our auditors, inspectors, right? Tell me the story and evidence to me that you can tell the story piece by piece by piece as we go through the documentation together. So it's very holistic view.
No, and I think the completeness will change with R3 in some sense, at least in how we will define it in some sense. I think it'll move from, today we're just checking some cases a minimal list of documents to meet it. I think we already today could start thinking about, when we have sites, we need this document. When we have investigator needs this. So you need, for this investigator, then you're complete when you have the financial disclosure, you have your CV, you have your training documentation. But with R3, we'll move into more risk-based. So we'll say, when we have these study risks identified, when have we mitigated that risk? And in some sense, I think longer term it'll become simpler, but it'll take time to change the mindset. But we'll say, okay, if you have a bipolar study, for instance, when we then select the investigator, it's not enough just to get your CV for this and med license for the investigator. We actually need to ensure that this investigator can assess a bipolar patient, right? So this is where we will go in and say, okay, are this investigator qualified to assess bipolar patients? And so in the TMF, think about, you will have a site, it's a study risk of bipolar, and now we need to ensure is the training there to assess bipolar patient. And now you have the documentation to support the risk. I think that's where we will go over the next 5 years. Is there more questions? Are we at the top of all hour?
Yeah, I think if Oliver, if you're on, should we continue with questions, or are we at our time? I think you're still on mute.
We can take maybe 2 more questions.
Okay.
Okay. So question has come here about, are there potentially better milestones for the extensive time between site start and database lock? For studies that we have that are extensive, multi-year studies, are there better ways to define milestones? Elvin, do you have any thoughts on that?
Yeah, we will have some time milestones. So there are some milestones already which are related to safety reporting and regulatory yearly reporting. They're normally yearly if we get into it. But in a preferred world, we have no milestones, because if the study just tugs along, it actually goes without problem. And the milestones will be when we do interim analysis. But these are normally not going into a TMF itself. They'll go into the EC and the data and analysis, but that's part of the bigger story, because it more becomes clinical endpoint driven. So it's often something we don't directly see in the TMF. But the only other things, when you get into the execution part, it becomes protocol deviations, it becomes protocol amendments. But in most cases, we're trying to avoid them, though there can be learning. So, but those become risks. So actually, I think study startup will end up behaving more like the execution, because it's more risk mitigation and documentation that we mitigated the risk. So I don't think we need more milestones. Preferably, we actually have as few as we can, because we're just tugging along and things go well.
Yeah, and I think just to add to that, I think it's maybe not so many milestones we need, but it's the adherence to the TMF filing, right? And making sure that we're following the milestones closely, assessing for completeness in an ongoing way, and making sure that our TMF is reflective of where we are at in the study life cycle.
PI change would be a thing you document again, right? But it comes into, you only file when you have a PI change. You shouldn't file because the PI changed their CV, because that's not a risk to the study. I think we need to have a reason for everything we file in the future, and not just file it because we receive it.
Yeah.
All right. So I said 2 questions, but I think we only had time for one. Elvin, if you wouldn't mind, stop sharing your screen. I'm just going to share 1 or 2 slides to finish off, just to let everybody know what's coming next. Thank you for that. And I'll add this to the stream. First of all, I want to thank both Elvin and Jennifer for such an amazing presentation. I saw a lot of questions coming in, a lot of chats. I think the audience really enjoyed the perspective. I too, like Jennifer, really enjoyed your analogy, Elvin, of the streets and the roads and the navigation to the cities and the counties. I thought that was a really neat way of explaining something that is obviously very, very complex. And so I think, you know, if we can simplify things, I think that's a path to reducing that complexity. So I want to thank you both. Of course, I want to thank everybody else for attending today's session. I do want to make note that we have a short networking break. So from 11 a.m. Eastern until around 12:30 Eastern. So you got about an hour and a half until our next session. Please head over to the networking booth. For those that have been asking questions, I think some of those folks are going to be in those lounges, sitting at tables. If you haven't gone there before, it's great. You can have a virtual conversation, a video conversation with 10, 15 different people around a subject. It's really, really fun. The next session is a panel discussion. It's titled Universe of Startup Records. How do we make it easier? So a really great follow-on from today's session. It's going to be chaired by Fran Ross, who's the TMF Practice Director at Advanced Clinical, who, of course, was a part of this session in the chat. And she will be joined by Radwa Ali from the George Washington University Medical Faculty, as well as Seneca Harrison from QCR and Etienne Hinton from Duke Clinical Research Institute. So they're going to be really giving that sort of site perspective, right? So we've heard a lot about sponsor startup, and we've heard a lot about some of the complexities there. We're going to be getting that perspective from the site in the next session at 12:30 Eastern. So don't miss that one. And as I mentioned, it is a networking. It's kind of a big deal. Go head over to the lounge, all of a virtual booth, chat with your peers and booth members on a range of subjects, meet new people. I'm there. I'll be chatting with you guys. There'll be a bunch of other people. Hopefully my fellow presenters today will join us in the lounge for some discussion, and we'll see you on the next session. Amazing. Thank you very much, everyone.
Thank you so much.
Just stopping the stream, guys.